The Betaproteobacteria refers to the group of bacteria that have a common function, that they usually use the nutrient substances that are released from the areas of anaerobic decomposition of the organic matter, such as H2 gas, hydrogen gas, ammonia, and methane. So I told you one of the member is Nitrosomonas. Another member is the Thiobacillus, and Thiobacillus is very much important, mostly in the sulfur cycles. So as I told you that there are different prefix and suffix that are used for describing either a process or a property. So same is the case with this word thio. It refers to, mostly it refers to the role in the sulfur cycle. Either it is the production of sulfur, or it is the reduction of sulfur, or it is the oxidation of sulfur. So mostly they are called as those activities are labeled as the word thio, right? So thiobacillus, actually they are the sulfur oxidizing bacteria. They are very much important in the sulfur cycle. So another question is that what is the importance of sulfur cycle in your lives? So sulfur is very much important as a functional group in the amino acids which are found in the proteins, right? So they are important because they are the part of the proteins playing very vital functions in your cells, right? And secondly, the sulfur cycle is maintained by these bacteria. How? Because these organisms, they are capable of obtaining energy by forming the by oxidizing the reduced form of sulfur like H2S and elemental sulfur, and they convert them into sulfates. So in usually sulfur cycle, it works in both the environment, in aerobic environment as well as in anaerobic environment, right? So whenever there is anaerobic condition, so H2S can be used as a replacement for H2O, and the oxidized form of sulfur can be used as a substitute, or you can say as a replacement for oxygen, right? So these organisms, they are mostly present in those areas which are anaerobic in nature. So in that areas, they are using the sulfur forms for maintaining their cycles. So this is one of the sulfur cycles, right? So you can find the role of microbial oxidation. There are different organisms we'll discuss later on, but targeting the role of Thiobacillus, right? So it can transform, it can use H2S, and it can transform it into either elemental sulfur, and it forms sulfates, right? So as you can see that there are different keys, right? So this green arrows, they are showing the oxidation. Now moving towards another member, that is the spirillum. So spirillum, as its name indicates, that it is some indicating towards those organisms that are spiral in shapes, and these spirals, they are also shown in the background, right? So there is another group of organism which is known as spirochetes. They also maintain spiral shapes, but there is a difference between spirillum and spirochetes. Spirochetes you have visualized in your first year staining labs, and the difference is in their motility. As you can see, that is spirillum, they are motile by means of a polar flagellum, a tuft of polar flagellum, right? So as you can see that these are the flagella that are shown here on both sides. They are highly motile, and they are motile by the presence of the polar flagella. The second thing is spirochetes. They are also motile, but their motility is maintained by endoflagella, right? So, or also known as axial filament. But in this group, this is not the case. They are motile by the polar flagella. They may be one or they may be in a group, right? So, secondly, their spirals, they are the loose spirals. Spirochetes, they are showing the condensed spirals, right? Their size is comparatively smaller than the spirillums, right? So, and they are mostly present in the fresh water, and they are aerobic bacteria, and they are mostly used as a demonstration slide for showing the motility pattern, and usually the Spirillum volutans is used in the labs to show the first year students the motility and how it is maintained, right? So since their shape is important, that's why it is being discussed here, right? Another important member is Burkholderia, and it was previously grouped with the genus Pseudomonas, but since you know that with the availability of 16S ribosomal DNA data, it is now regrouped into the Betaproteobacteria, and they are motile again by the flagella and the tuft of flagella. And why this group is important? It is important because it has some exceptional nutritional spectrum, that it is capable of degrading more than 100 different organic molecules. So its degradation pattern, its metabolic activity is very diverse. It can degrade so many organic molecules, right? And best known species and best studied species is the Burkholderia, Cepacia, or Cepacia, you can also say this like. And since if any organism is capable of degrading so many compounds, means a wide range of compounds, so we can believe that it may contaminate the places where very limited number of nutrients are there, or a unique combination of nutrients are there, right? So as you can see that because of this capability of degrading vast number of organic molecules, this particular organism has a very sound significance in the hospital environment, that it can contaminate the equipments, it can contaminate the drugs, and it actually can grow in the disinfectant solutions. So usually we disinfect the things to prevent infections, but if someone can grow in the disinfectant solution, so that's why it needs to be discussed, that there are some organisms which are so unique in their microbial degradation pattern that they can degrade wide variety of organic compounds, that they can even grow in the disinfectant solutions. I'm not talking about survival, I'm talking about their growth, right? So this is very unique in that sense, right? So, and secondly, this particular organism is or this group and the members of this group is very much important for some patients which have a lung disorder that is known as cystic fibrosis grow there, and it can further complicate it, right? Complicate the disease and further enhances the complications of the disease. So moving towards the next genus, that is Bordetella, and Bordetella is again gram-negative rods, and they are non-motile and they are aerobic, and one of the prominent member of this genus is Bordetella pertussis, and The species Bordetella pertussis, responsible of causing an infection known as pertussis, and it is also known as whooping cough, right? So this is known for and this particular infection targeting mostly children. Neisseria, I hope that you have heard about Neisseria in your intermediate classes, because it is one of the bacterial pathogen responsible for sexually transmitted disease. And Neisseria is unique in the sense that so far you have not studied gram-negative cocci. So they are the gram-negative cocci, which are motile as well, and they are aerobic in nature. So this is unique in their morphology, that they are gram-negatives, but they are cocci. So it is first type of this genus we are studying here, which is cocci in shape, and gram reaction is gram-negative. So they are the normal inhabitants of the mucous membranes of mammals. And the genus Neisseria is commonly present in the mucous membranes of mammals, but a few members, they are also pathogenic as well. So the dominant, or you can say the famous pathogenic species include, I hope you know the names: Neisseria gonorrhea, which is a causative agent of gonorrhea, and another important pathogen is Neisseria meningititis, right? And it causes meningococcal meningitis. Now, meningitis is the infection of meninges. What are meninges? Meninges are the layers surrounding your nervous system, protective layers, right? 3 layers, they are called as meninges. So the Neisseria meningititis, it targets those meninges, and it causes meningitis, and it is one of the serious infections targeting again the children. Zoogloea, it's another genus, gram-negative organisms, and as you have heard about Zoogloea, that it is involved in sewage treatment process, and they are aerobic in nature. And basically, in these organisms, the members of these organisms, they form a thin film on and slimy film on the surfaces, and which is responsible for the degradation of the organic matter present in the sewage water, right? So they are important with their ability to degrade the organic matter, and they form a bacterial film, which is known as Zoogloea, right? So the members of this genus, they are grouped into this category. So without zoogloeal films, these sewerage systems, based on these films, they do not operate functionally.